our first speaker opening the webinar with a general introduction on the progression of methods in cancer research, Professor Marianna Kruithof-de Julio, who is a leading researcher at the University of Bern and directs the Urology Research Lab and Organoid Core Facility for advancing precision medicine in urology. Her team has pioneered patient-derived organoids for bladder, prostate, and pancreatic cancer, alongside microvascular on-chip chambers to study single-cell function. By exploring tumor cells, trauma, immune cells, and vasculature together, her research offers a holistic view of cancer progression, and she focuses on addressing this way clinical challenges. Generally, her work aims to bridge research and patient care with the goal of predicting and controlling metastases for improved patient outcomes. So I'm happy to turn the presentation over to you, Marianna, and we are really looking forward to your talk.
All right, so thank you very much for the very kind introduction, and I'm really glad to be here and part of this today. I was tasked with giving you an idea of what the progression of methods in cancer research are. So I took this task at heart and started a bit far in 3000 before Christ in the timeline of what is the study of cancer, all the way to coming to modern oncology. And what is quite remarkable about this is, of course, that the first few years of us studying cancer were really not much going on. But there was this development of treatment that was focused mostly on what at the time they were doing as surgery. The really first change and the very first impact was, of course, in the beginning of the 1900s with Marie and Pierre Curie, of course, when they started treating with using X-rays. Until the first chemotherapeutic was developed in 1940. And this was really a landmark change in which many of the cancers were treated, of course, with platins and chemotherapeutics. Interestingly enough, this was still, let's say, the era in which being things discovered bit by bit, everybody was being treated in the same way. It was only in the 1980s that we started with the more targeted therapies. And this is really the onset of what then 40 years later we call precision medicine or precision oncology. And these were the discovery and the treatment with the tyrosine kinase and the inhibitors and the different monoclonal antibodies. And then finally, we arrive in the 2010s, in which the checkpoint inhibitors have been discovered, and immunotherapy has started to take a bigger role in the treatment of cancers. Although I must say that in the case, for example, of bladder cancers, a sort of immunotherapy has been going on already for quite a long time with the BCG treatment. But now if we focus it down and narrow it down to, let's say, the more modern times, or the last if we can say 10 to 14 years, as you can see, there is a whole amount of new discoveries and new focusing, in particular in the area of immunotherapy. And this is actually really good news, because there's really a lot of treatments of cancers that are much more treatable in a respect. But again, one of the tasks, so the task that I had was to tell you the good, the bad, and the ugly of the models of cancer. So one of the major models to study cancer in the lab, of course, is the 2D cell culture. And this is one of the major models that has been used. Independently, we're talking now, of course, of cancer of origin and cell type. But the advantage is, of course, that the cells here are of human origin. They're some genetically stable, but most importantly, they're easy to handle. There's unlimited growth, and they're relatively cheap. Of course, there's plenty of disadvantages that go with this. And one of them would be, for example, the loss in the absence of the microenvironment. So slowly in the years, things became more complex. And one of the things that people have been using a lot are GEMs, so genetically engineered mice models. Genetically engineered mice models have had a great success in the study of cancer and cancer progression, because one can imagine to modify one gene and then to study the cancer in its progression in an animal. And of course, there's presence of the complex physiological animal in there, and the heterogeneity of the populations, but of course, the stroma and the immune cells of the animals itself. And of course, this being the advantage is also the disadvantage, because the stromal murine is from a murine contribution, of course. The other, of course, disadvantage is the high costs that are associated with these animals, with the animal use, and of course, the time-consuming in terms of developing of the model itself. And of course, the fact that we're working with animals itself. Another concept of animal working is the patient-derived xenograft. These are not genetically engineered models, so these animals do not have an immune system, or at least don't have a full immune system, and this is definitely a negative aspect of these models. However, the genetic stability and the fact that the tumor grows of human origin indefinitely in an animal, and allows then the study and the monitoring of response to it in vivo, is definitely a plus. Of course, in the years, for many reasons, one of them is also the more consciousness of the fact that we are working with live animals, has then stirred the research community to look for other models. Before we touch that particular base, one in between step can be considered, for example, the generation from patient-derived xenograft of ex vivo tissue slices, meaning that the tumor is then studied in a dish and not studied in the animal. So the selection, for example, of a large number of drugs doesn't have to be done anymore in a large animal experiment, but we can reduce the amount of animals if we do a first passage on ex vivo tissue slices and narrow down the compounds that we think might be of particular interest in vivo in this way. The next step, of course, was to really move away from the animal models. And one of the things that, is been a hype in the last maybe 10, 15 years is the generation of organoids. Now, be it as it may, we can debate the word organoids and what we mean for it. But for the purpose of this study here, I would like to define an organoid as a multicellular structure, and that can be derived from adult organism, that can be derived from pluripotent stem cells. And in its early version, an organoid would range simply from epithelial cell structures, whereas now what we are talking about is also more multiple and complex structures that have incorporated stroma cells and immune cells. Again, I think that the semantics and of the word organoid is still debatable, but in this case, let's take it with a grain of salt. There's a very interesting, I would say, timeline in the case of organoids, and actually, this is something that I was very interested in, because if you look at this timeline, the organoids it's not an event that has been found in the last, or a structure that we started studying in the last 10 years, but actually it's already from the beginning of last century that people have been looking how to complexly arrange in a three-dimensional structure cells. And the concept basically is that what we want to do is that we want the cells to interact with each other. And this interaction will allow a better understanding of what the what the signals are that allow the cells to, for example, proliferate. There's many examples that we could show you about organoids. This is work from one of my ex-PhD students, which has successfully defended her PhD, Martina Minoli. And here, what she shows is the generation of organoids from bladder cancer. I think what is very interesting about this particular picture is that you can see the concept of heterogeneity of the cancer itself, but not only of the cancer, of how the organoids assemble and form with each other. Many techniques and technologies have been developed in the process because, of course, it's very important we can characterize the organoids. And one of the things that we do is that we embed them and in drops of, for example, in this case, it's hydrogel. We cut them and we can, of course, stain them. In this case, you see a representative H&E staining, but of course, immunofluorescence and immunohistochemistry can be performed on these organoids. The other concept of the organoids, of course, is the fact that they are growing in matrigel. So a lot of systems do not allow the formation of these organoids without the matrigel. For bladder and prostate cancer, we have identified already a few years ago a system, and it instead does allow us to grow the organoids without matrigel. So we can grow them in ultra-low attachment plates or in these plates from this company, Krugenmeier, which allows us basically to visualize them individually. And what is really particular is that if you look at these videos here, you will see the time lapse of an organoid that is starting to form from an aggregation of cells. And this allows us the study of the process of aggregation and what those signalings are that are associated with the organoid formation. But if you look at the other video, what is also fascinating is this structure, this organoid that is actually in theory and keeps on developing and expelling or incorporating cells as it grows. So this system that we have generated without matrigel, in fact, allows this visualization of the individual organoids and the better understanding of how, for example, a drug or a chemotherapeutic would function and interact with the organoid itself. The other importance is the penetrance of the drug to the organoid, and this is also one of the reasons that in drug screens one might want to consider the dimension and the homogeneity of the size of the organoids in its culture. Speaking about a drug screen, this is an example of how this can happen. This is, for example, how we follow a organoid from a cell line that's called an RT4. This is a cell line that has a mutation, so it responds to the erdafitinib. And what you do see here is that in conditions like mitomycin and gemcitabine and docetaxel, at day 0, the organoids have formed, and after 48 hours of the drugs, there seems to be no effect. That these organoids are matching, so at day 0 and day 2. Whereas in the case of the erdafitinib, but also in the case of ebirubicin and our control puromycin, instead, you do see a morphological effect on the organoid that is visual here, which helps a lot in how we can study the cancer. With that said, mice are not only the only animals that are used to study cancer. In particular, there's also zebrafish. Zebrafish models can be used at different stages. In this case, these are embryonic stages. And what we do is that we inject in the ductal clavir the cells, and then the cells go in circulation. And here, as you can see, the fish vessels are highlighted in our GFP positive, whereas the cells that we have injected are red. And what you see is that if you zoom in the caudal area, which is in general the area where there is the extravasation, And if you look at this matrix that is around here, this is the collagen that is the fin of the mouse. You will see the red cells that leave the vascularization and enter the collagen in a sort of displacement kind of manner. What you would see here, if you would run a 2-photon, you would see the collagen really moving and being displaced for the cells. We can use light sheet microscopy to reconstruct three-dimensionally the vessels and this area or the entire fish. That is the advantage of using embryos of zebrafish. And what you can see here is that, again, green is the vascularization, red are the cells, and you will be able to catch the extravasation of these cells. And of course, you can imagine that maybe if you're interested, you can genetically modify the cells and see if your favorite gene has an effect on extravasation, or maybe use a compound. Using compounds in zebrafish, you have to be very mindful because the zebrafish are at an early stage of development. So the compound that you might be using might be interfering with the development itself of the fish. Of course, zebrafish is, of course, an animal as well. So one of the other things that we have done, we as a scientific community, we have moved away and looked more also on solutions that are organ on chip. I think this is a very nice review that shows the different methodologies and the different kind of experiments that can be used. And on-chip system is, well, not as straightforward. You can be the end user, which is the person that actually uses the chip, but the whole process of fabrication and development of the chip requires different set of skills, at least that I have. So one of the things that we have done is that we went to the engineers in our talk, in particular, Professor Winan, and he has developed a microvascular on-chip system, which is a chip that has several side chambers, flow chambers in the middle that are pointed here with red arrows, and the central chamber, which is highlighted from the green arrows. And if you look at the video, what you will see is that in cells that are flowing from the side chambers through the pillars that are created from the network of HUVEC cells that generate the vascularization and enter the chamber through the openings. So it's a little bit of a fast video, but what you see here is exactly that the cells are entering basically the system and walk around in this network of vascularization. This, of course, is a very interesting methodology that allows us to study how the cells might interact with the vasculature and what potentials do the cells have to extravasate. In particular, I will show you a closer example of this. In what we do, confocal microscopy, and you look at the cells that are inside the vascular chamber, you see that some of the cells tend to try to escape the system. So if you look carefully, this gives you basically an idea of this one cell that's leaving. And now if you have a look at the cells that are here in this circle area, you will see the cells that leave in real time the vasculature and then decide to move back inside. Of course, what I've told you about now is all systems that are in vitro and ex vivo as well, and in vivo, but there's also a huge impact also of cancer models that come from data-driven approaches and that are basically in silico. So I also, this particular review here shows you the huge increment in the years of papers published that use cancer modeling in silico. And really, one of the things that have been going on is that cells and cancer has been studied, but not only cancer, of course, at different levels. So starting from a gene level to a protein level to a cell level, and then slowly going in these more complex models that allows the looking at the system from also a spatial point of view, from tissue level. And this hopefully at one point will bring information at both organs level, and then patient, and finally population, to better understand and to finally maybe stratify patients in a better and different way in terms of how they respond to treatment. So now the question, what's next? Well, is CRISPR and the future of cancer, is immunotherapy going to boost cancer? How are the future diagnoses going to be? Is virtual biopsy going to be the new concept? How are we going to talk about precision medicine? Does precision oncology even exist? And also another very important aspect is where do the cancer vaccines stand in all this? And finally, I would like to leave you with a question: is this future that we're looking at, will this render the animal models obsolete? And with this, I would like to thank all of you for your attention and being here today, and of course, my group for working hard in this fight of cancer. So thank you very much.